Eukaryotic DNA, as stated previously, is much more complex. Eukaryotic DNA consists of linear chromosomes that are commonly packaged with proteins called histones. Together, this forms chromatin. When the DNA needs to be wrapped up, it will form what is termed a nucleosome. A nucleosome consists of one unit of histone proteins with the associated DNA wrapped around it. This resembles beads on a string. Keep in mind that the DNA is considered to be an acid, nucleic acid. As such, it is negatively charged. Remember that there are many phosphate groups with many oxygens with a negative charge that exist in the backbone of the DNA molecule. Histones are protein-based and are considered to be basic and thus positively charged. This positive-negative interaction between the DNA and the histones is very important in wrapping and unwrapping the DNA molecule. How tightly the DNA ends up wrapped around the histone protein will dictate how accessible it is to the process of transcription and translation. We usually term accessible DNA as euchromatin. However, if the chromatin ends up being wound up very, very tightly, to the point that it is inaccessible and considered to be silent, we would then term that DNA as heterochromatin. In prokaryotic cells, most of the DNA will code for proteins, or at least will be coding for RNA molecules. However, in eukaryotic cells, we know that almost 97% of eukaryotic DNA is considered to be non-coding. However, this can include many different important regions of the DNA, such as introns, promoter regions, enhancer regions, terminator regions, centromeres and telomeres, as well as repetitive DNA sequence, to which the function of these sequences is yet unknown. There are many different options to controlling gene expression in eukaryotic cells. Once again, this is typically a reflection of the complexity of their genome. When we're talking about regulating gene expression, one of the important ways that eukaryotic cells can regulate their genes is by changing the overall structure of the chromatin. This involves winding and unwinding the DNA to make it inaccessible as well as accessible for transcription and thus translation. Here we have the interaction between histones and the DNA. Histone proteins can be modified to make the DNA wrap more or less around them. Once again, if the DNA is more tightly bound, it will be considered silent or inaccessible for transcription. DNA that is unwound around the histone is considered to be more accessible. Histones can be both acetylated or methylated in order to wrap and unwrap the DNA. By adding acetyl groups or 2 carbon groups to the histone proteins, this will act to loosen up or reduce the connection between the DNA-histone interaction. Thus, it will loosen the DNA, making it more accessible for transcription. This, in essence, will allow for the gene to be either turned on or upregulated. We can also methylate the histones. In this case, a 1-carbon methyl group will be added to the histone. This can also be used to either positively or negatively affect transcription rates. Keep in mind that the DNA itself can also be methylated. In this instance, the methyl groups will be added to the cytosine residues on the DNA nucleic acid. This will help to convert regions of the genome into heterochromatin, thus making them inaccessible or silenced for transcription and translation. Examples of instances where the DNA itself is methylated involve bar bodies, centromeres, as well as telomere regions. Thus, the balance between histone acetylation and histone methylation will help to wrap and unwrap the DNA to control gene expression. In addition, DNA methylation can be used to form heterochromatin to silence complete areas of the genome. This gives an overview of the wrapping and unwrapping of DNA. Note that this is working at the level of controlling gene expression at the level of DNA. Note that in the top right of this diagram, we have an example of what centromeric DNA would look like. Note that there is much folding around the multiple histone proteins, essentially forming an area of heterochromatin located at the centromere region. This allows for the kinetochores to be present and allows for the chromosomes to be separated without tearing at the extreme ends. Note in the diagram below we have an example of DNA acetylation. Note that a histone protein form a complex that then allows for the DNA to wrap around it. Keep in mind the histone proteins are considered positively charged, whereas the DNA itself is considered to be negatively charged. One unit of histone proteins with its associated DNA is termed a nucleosome, which we can see in the diagram below. In nucleosome formation, we can see that the histone tails protrude outward from the nucleosome center. It is on these tails that we have specific amino acids that are available to be modified either through acetylation or methylation. We can see that when histone tails are unacetylated, they form compact DNA. When the DNA is in this form, it is not available or accessible for transcription. However, as acetyl groups are added to the histone tails, this will interrupt the interaction between the histones and the DNA, thus allowing for the DNA to unfold and become accessible for transcription. In many other organisms, there are multiple levels of gene expression control. One of the important things to recognize in many multicellular organisms is the fact that every single cell in that organism contains the exact same genome. However, different cells can express different genes. They can do them at different times or they can do them at different levels. This can be termed differential gene expression. The way this is carried out is by having specific regulatory sequences of DNA that will help control rates of transcription. This can be done through the promoter enhancer regions as well as changing chromatin structure. One can also start and stop and regulate the rate of transcription. In this level of control, we have proteins that are called transcription factors. Transcription factors are important to binding to specific regions on the DNA, such as promoter regions or enhancer regions, and they will be acting to either turn transcription on or off, and once again, they can also upregulate or downregulate the rate of transcription, thus affecting and controlling gene expression. This is an example of gene control and gene expression at the transcriptional level. Keep in mind that there are many other mechanisms to also control gene expression post-transcriptionally, which we will take a look at in the next few slides. This slide here gives an overview of the action of transcription factors. Keep in mind that in the above example, we have DNA that is present in all cells. In this example, we have the DNA coding regions for both the albumin gene as well as the crystalline gene. However, the albumin gene will be important for liver cells to produce, whereas the lens cells would need to produce the crystalline gene. Let's take a look at how these different cells can activate different genes depending upon their specific needs, even though they consist of the same genetic information. With respect to the DNA sequence, we can see that there are enhancer regions, one for the albumin gene, which lays above the promoter region and the gene of interest. Note that we also have crystalline enhancer genes as well. Once again, these enhancer regions will lie upstream of the promoter region as well as the gene of interest. In the liver cell, we can see that we have specific activators that are produced for activating the albumin gene in this instance. These activators, once produced, will bind to the enhancer regions for the albumin gene. This will then cause a conformational change in the DNA to make it more accessible for gene transcription. In this instance, we would also have transcription factors that would help to bind to the promoter region. This will allow for RNA polymerase to recognize the region on DNA that contains the gene of interest. This would direct RNA polymerase to bind to the specific promoter. As a result, it will be able to access the albumin gene. As a result, this gene would be expressed, meaning that an RNA transcript would be made and the subsequent proteins would also be formed. Note that the crystalline gene in this cell type does not have any enhancers activated. Thus, this gene is not expressed or it is silent. Now let's take a look at what is occurring in the lens cell. In the lens cell, we would now have specialized activators that would be specific for the crystalline gene. In essence, they would be binding to the specific enhancer region on the genome. Transcription factors would also bind, thus enhancing transcription of the crystalline gene. Note that these activators are not able to bind or fit in the enhancer region for the albumin gene. As a result, the albumin gene will not be expressed. This illustrates one of the most important ways that eukaryotic cells can express different proteins in different cell types. This also helps in the differentiation of cells as well, meaning that during embryogenesis, certain cells will be differentiated into specific cell types. One of the ways in which they do that is by producing specific materials that will activate specific genes that are important for their overall function. We are now going to take a look at transcriptional control. Every gene has a unique and very complex combination of various control elements. Transcription factors in general have a DNA binding site available on themselves. This binding site will help them to recognize their specific control element or control sequence on the DNA. Transcription factors, once again, can affect transcription in many ways. They can recruit or block RNA polymerase from binding and accessing the DNA. This can be done by forming a transcription complex. Transcription factors can also work in altering chromatin structure, making DNA either more or less accessible to transcription and translation. Finally, different cells can produce different types of transcription factors. This is an important step in being able to, once again, have differential gene expression. This diagram here gives you an overview of the action of transcription factors. Note that in our DNA sequence, we once again have an enhancer region which lays upstream or before our gene of interest. Here we call this a distal control element. It is controlling a gene of interest further along or far away from the enhancer region. Note that we also have activator proteins that would be produced by the cell. These activator proteins would join to the enhancer regions. This would act to help recruit transcription factors to the area. When transcription factors bind, they can form a transcriptional complex. In this instance here, this complex exists between transcription factors, mediator proteins, activator proteins, as well as a DNA bending protein. Together, all of these components would join to change the overall structure of the DNA. In this case, it would make it more accessible to gene transcription. Once the complex forms, RNA polymerase can locate the correct promoter region. Once it binds to the promoter, it can then access the genes of interest to start and initiate transcription and eventually translation. Note that within eukaryotic cells, we have seen transcriptional control as well as pre-transcriptional control. There can also be levels of control that can be utilized after transcription. These can exist as post-translational or pre-translational levels of control. One of the ways that the cell can control what is expressed after a transcript has been made involves alternative RNA splicing. Some genes can lead to the production of various types of proteins, and that is dependent on how the exons are spliced together in the final mature mRNA molecule. For instance, some exons can be skipped and others can be added. The difference in the combination of exons will lead to the production of different proteins. Another way in which eukaryotic cells can control gene expression is by looking at the level of the protein itself. Proteins can be stabilized or degraded, and this will help it control gene expression. If a protein is no longer needed in the cell, it can be targeted for degradation. This is usually done by adding a ubiquitin protein. Once the ubiquitin is tagged to the protein, this complex can then be targeted for digestion in the cell. One of the organelles that helps to perform this function is the proteasome. MicroRNA and its use in the cell can also control gene expression. MicroRNA refers to small RNA molecules. These molecules do not code for any protein, however, they do have an important role in controlling gene expression. In this instance, they will block the translation of a target mRNA molecule. They will also target it for degradation. As a result, these microRNAs will act as small tags to notify the cell that this mRNA transcript is no longer needed. Overall, there are many different layers of regulation that can allow for eukaryotic cells to fine-tune and control their gene expression. This diagram here illustrates the concept of alternative RNA splicing as well as the role of microRNAs. In the top diagram, we have a DNA molecule that consists of 5 specific exons. These exons are part of the troponin T gene. We can see that the primary transcript that would result from this DNA template consists of all 5 exons. However, we can see that there are multiple ways to splice this RNA molecule. In one instance, we have the sequence of exons being 1, 2, 3, 5. This would lead to the production of one specific protein. However, if different exons are kept, such as 1, 2, 4, 5, this would lead to the production of a different protein. Thus, we can have the production of multiple different proteins using one primary transcript, and this is simply dependent upon the type of exon that is kept. In the bottom diagram, we can see the role of microRNAs. MicroRNAs are a small RNA sequence that are important in identifying and binding to a target mRNA sequence that will target it for degradation. They can also block the translation of that particular RNA transcript. We see that the microRNA will join to a protein to form a microRNA protein complex. This protein will help the microRNA to locate the appropriate target mRNA sequence. Once the microRNA and protein complex bind to the target mRNA, this could denote the mRNA for being tagged for degradation. In this instance, the target mRNA would be degraded and inaccessible for translation. However, in some instances, the binding of the microRNA protein complex can halt or prevent translation from occurring. In this instance, the RNA transcript would still be present within the cell, however, it would be silenced. If the protein needed to be translated, this microRNA protein complex could release the target mRNA molecule, at which point it would then be accessible for translation. Finally, this slide gives an overview of the multiple different mechanisms that eukaryotic cells can utilize in order to control their gene expression. As you can see, it can be exhibited at multiple levels. Eukaryotic cells can change the structure of their chromatin to unwind and wind it to make it more or less accessible for transcription and translation. They can further modify the DNA by using DNA methylation to permanently silence regions of DNA. Eukaryotic cells can also use techniques to control transcription and translation at the level of transcription. This involves alternative RNA processing, as we've seen in the slide before. Keep in mind that the RNA in eukaryotic cells has to be spliced properly, as well as caps and tails, 5' G-cap and 3' poly A tail must be added to the RNA sequence in order for it to leave the nucleus. Once the RNA leaves the nucleus, there are still mechanisms that can be used to control gene expression. These include using microRNAs to tag the RNA for degradation or to block translation from occurring. And furthermore, gene expression control can also be employed at the level of the protein itself. This can be done by stabilizing a protein to maintain its activity or by tagging it with ubiquitin to cause it to be degraded and non-functional in the cell. Thus, we can see that the control of gene expression in eukaryotic cells is a much more complicated process than we've seen in prokaryotes.